this video, we're going to talk about the machinery needed to do intron splicing, and that's called the spliceosome. Now, inside the eukaryotic nucleus, you have a lot of copies of highly conserved 60 to 300 nucleotide RNAs that are called small nuclear RNAs or snRNAs. These snRNAs will combine with proteins and you'll make a complex called small nuclear ribonucleotide proteins or SNRPs. One example is the U1 SNRP, and it's called U because there's a lot of uridine in it. And this will recognize the 5' splicing junction that we talked about in the previous video. And there's a lot of other proteins as well, or SNRPs rather, U2, U4, U6, U5. And this splicing happens in a large particle, 60S, called the spliceosome. And the spliceosome will bring together the pre-mRNA, the SNRPs, and all these pre-mRNA binding proteins to get rid of the introns. And this is probably the most complicated complex that we know of in the cells to modify the introns of your mRNAs. Now let's take a look at these SNRPs. And these SNRPs have, all of the SNRPs have a core protein, a SNRP core protein. Now 7 SM proteins are named B, D1, D2, D3, E, F, and G. And these proteins will bind to a conserved sequence, AA, UUU, GU, GG. This is known as the smRNA motif. And we see this motif in U1, U2, U4, and U5 small nuclear RNAs. Now these proteins will form a 7-protein ring, a heptatomeric ring. And in the center of this ring, you have a positive amino acids. And it's just large enough for one of your small nuclear RNAs to fit in. So you're forming this ring, you put your snRNA in the center of this ring, and now you have your ribonucleotide protein ready to go. Let's take a look at the U1 snRNA, which is, the U1 SNRP is made out of U1 snRNA and 10 different proteins. What we're showing right here, though, is just the U1 snRNA. And this is how we typically look at RNA to try and determine the structure of it. And this is a guess for the most part. It's very hard to get an actual structure of RNA because they're so flexible. So the techniques we have currently at our disposal are good for proteins, horrible for RNA. So this is usually made by a computer program that will tell us what we think the RNA looks like. And also, this is a 2D image. Keep in mind, this is 3D, so this can be twisted on itself a lot of different ways. But what we think the U1 sRNA looks like is that it has 5 double helical stems. And 4 of the stems come together at a 4-way junction. So that's what's being shown here. You have a 4-way junction forming for your stems. You can see here's our smRNA motif, our AUUUGUG. This is where our proteins can bind. And we have a 5' splice recognition motif where we'll find the 5' end of the intron so we can cut it out. Now, why have introns at all? Well, 2 important roles for introns. 1, protein evolution. So we've been talking about domains of proteins and how you can make different proteins with different domains. If you just swap out domains, you can make a whole new function. Well, the idea is the same with exons. You can make proteins by just having a collection of exons that are assembled by recombination of neighboring introns. So each exon's like a different domain or different motif. you put these motifs together and just have them separated by introns, now you can mix and match your motifs and you can have a protein. That's really the second advantage. That this splicing allows a single gene to encode a lot of different proteins. So if you mix and match how your exons are arranged, you now have proteins with slightly different functions. You're not going to get anything too crazy because you're still using the same parts, the same motifs, but you could work on slightly different substrates, for an example. And so let's take a look at this alternative splicing. And a lot of genes will go through alternative splicing. So if you have more than one exon, you can put the exons together to make slightly different proteins. For example, there's a rat gene that this gene is spliced differently 7 different ways based on what tissue it's in. So that's what's being shown here, where the gray parts are the introns, the colored part are the exons, and depending on what type of tissue this gene is in the rat, it'll have different exons that are used to express this protein. And this alternative splicing happens in all multicellular organisms, especially vertebrates. 95% of our structural genes have alternative splicing event. And each structural gene inside the human body roughly codes for 3 different proteins. And so that's kind of what makes it hard looking at DNA between different organisms and trying to say which one's more complex, because the number of genes doesn't say how many proteins you have. With alternative splicing, as we see here, one gene is 8 different proteins, or 7 different proteins rather. So we can't really determine protein diversity by just looking at DNA. And here is another way that alternative splicing is important, and one way you can determine which introns to choose and which ones not to choose. And we're going to look at the transformer gene that is used in sex determination of Drosophila. So if you're not sure what Drosophila are, they're like little fruit flies, and they are used extensively in biology and biochemistry as model organism. Now, if we look at the exon 2 of TRA, there are 2 different splice sites we can choose from. The proximal splice site is used in males, while the distal one is used in females. So how can we determine which one to use in males and females? What is also interesting is that between these 2 sites, there is a stop codon. So in males, the UN SNRP auxiliary factor, which is called U2AF, will bind to the proximal 3' splice site as being shown here. That will mean that the mRNA will produce this stop codon. And when it makes this stop codon, this UAG, this protein is non-functional. So males don't have this TRA protein. While in females, they have a specific protein that is only made in females called SXL. This SXL binds to the site, the 3' proximal splice site, where U2AF binds in males. Since U2AF cannot bind here, it will bind at another place. This will cause a functional TRA protein, and that's kind of what these lines are showing, right? So this line showing I'm going to connect 1 to blue. My blue has my stop. Here with SXL, this U2AF is going to choose this splice site instead. So I'm going to connect 1 to 2 and 2 to 3. I am going to chop out this premature stop codon. So this gray bar is showing an intron. This blue is an exon. Males cut right here in the intron because of U2AF. That's what's doing the cutting for us. SXL blocks U2AF from binding there. So instead, we cut right here at this junction, knock out the premature stop. And we will see, or we have seen similar mechanisms for repressing or activating various splice sites in vertebrates. And in fact, 15% of genetic diseases in humans are just point mutations that will cause alternative splicing defects. So it's just from introns cutting that causes 15% of human genetic diseases. And that's it for this video. I will see you in the next video.